Welcome to the channel of Pharma. Today we see the ICS guideline on M7R2, that is assessment and control of DNA reactive mutagenic impurities in pharmaceutical to limit potential carcinogenic risk. It is published on April 2023. This is a documentation revision history. Latest guide is published in April 2023. This is a table of content. The introduction, the synthesis of drug substances involves the use of reactive chemicals, reagents, solvents, catalysts, other processing it. As a result of chemical synthesis or subsequent degradation, impurities reside in all drug substances and associated drug products. While ICSQ3A are two impurities in drug substance and ICSQ3B are two impurities in new drug products, provides some guidance to qualification control for the majority of the impurities. Limited guidelines to order for the impurities that DNA reactive. The purpose of this guideline is to provide practical framework that is applicable to the identification, categorization, qualification, and control of these mutagenic impurities to limit potential carcinogenic risk. This guideline is intended to comply to the ICS Q3A R2, ICS 3B R2, then ICH M3 R2 non-clinical safety studies of the conduct of human clinical trials and marketing authorization for pharmaceuticals. The guideline is emphasize the consideration both safety and quality risk management in emphasizing level of mutagenic impurities that are expected to pose negligible carcinogenic risk. It outlines recommendation for assessment and control of mutagenic impurities that reside the reasonably expected to reside in the final drug substance or product taken into consideration for intended consideration for human use. Scope of guidelines. This guideline is intended to provide guidance for the new drug substance and new drug products during their clinical development subsequent application for marketing. It also applies to most approved submission of marketed products to a new marketing applications for products with a drug substance that is present in the previously approved drug product. In both cases, whether changes to the drug substance synthesis result in a new impurities or increase acceptance criteria for existing impurities. Changes in formulation, composition, manufacturing process result in a new degradation products, increase acceptance criteria for existing degradation products. Changes in indication and dosing regimen are made which significantly affect the acceptable concern risk level. Assessment of the mutagenic potential of impurities is described in the guideline is not intended for the following types of drug substance and drugs products. That is a biological, biotechnological, peptide, oligonucleotide, radiopharmaceutical, then fermentation products, herbal products, crude products of animal and plant origin. This guideline is not applied to the substances drugs product intended for advanced indication that depend on the purpose of the ICH N9. S9. Additional, there may be some cases where drug substance is intended for the other indication. It itself generate toxic at therapeutic concentration may be expected to be associated with the increased cancer risk. Exposure to the mutagenic impurity in these cases would not be significantly added to the cancer risk of the drug substance. Therefore, impurities could be controlled at acceptable for non-mutagenic impurities. Assessment of mutagenic potential for impurities is as described in the guideline is not intended for the exhibit used in a existing marketed products, flavoring agents, colorants, perfumes. Application of this guideline is to leachables associated with the drug product packaging is not intended, but safety risk assessment principle outlined in this guideline is limiting the potential carcinogens that can be if warranted. The safety risk assessment principle of this guideline can be used if warranted for impurities in experiments that are used for the first time in the drug product and chemical synthesis. General principles are mentioned in this chapter. The focus of the guideline is the DNA reactive substances that are potentially how protein directly cause DNA damage when present in a low levels, leading to the mutation and therefore potentially causing cancer. This type of mutagenic carcinogen is usually detected in a bacterial reverse mutation. In mutagenicity assay, other types of genotoxicants that are not mutagenic typically have threshold mechanisms usually do not pose carcinogens in human risk at the level ordinarily present in the impurity. Therefore, to limit the possible human carcinogenic risk associated with the exposure to the potentially mutagenic impurities, the bacterial mutagenicity assay is used to assess the mutagenic potential that need to control structure-based assessment are useful to predicting the bacterial mutagenicity outcome based on the established knowledge. They are relative approaches to conduct the evaluation of a review of the chemical literature and computational toxicological assessment. A threshold of toxicology concern TTC concept was developed to define an acceptable intake for any unstudied chemical that poses negligible risk of carcinogenicity or other toxic effects. The methods often used the TTC is based on the generally considered as to be very conservative since in all the simple linear exploitation from the dose giving a 50% of tumor incidence at TD50 in a 1 of 10 is incidence using TD50 data for most sensitive site of tumor indication for application of a TCC in the assessment of applicable limit of mutagenic impurities in drug substances or drug products value of 1.5 microgram day corresponding to theoretical 10 to 5 excess lifetime risk of cancer can be justified. Some structural groups were identified to be such high potency that makes even below the TTC will be theoretically associated with the potential for a significant carcinogenic risk. This group is a highly pathogenic portion reported as a comprehensive alpha toxin like N-nitroso, and alkyl compounds. During clinical development, it is expected that control strategies and approaches will be less developed and earlier phases where overall development experience is limited. This guideline has acceptable index for mutagenic impurities on established risk assessment strategies. Acceptable risk during the early development phase is set to a theoretically calculated level of approximately one additional cancer per million. For later stages development and for marketed products, acceptable increased cancer risk is said to be theoretically calculated level of approximately 1 in 100,000. The risk levels represent the small theoretical increase in risk when compared to the human overall lifetime experience of developing any type of cancer, which is greater than 1 in 3. It is noted that established cancer risk assessment is based on the lifetime exposures, that is, less than lifetime exposures, both during development and marketing can be higher acceptable index of impurities and still maintain comparable risk level. The use of numerical cancer risk value 1 in a 1 lakh and its transition into risk-based dosage DTC is a highly categorical concept and should not be regarded as a realistic indication of the actual risk. Nevertheless, the TCT concept provides an estimate of self-exposure to any mutagenic compound. Exceeding the TTC is not necessarily still associated with the increased cancer risk, even in the conservative as assumption employing the duration of the TTC value. The most likely increase in a cancer incidence is actually much less than 1 in a 1 lakh. In the addition, in cases where mutagenic compound is non-carcinogenic is a rodent bias, there will be non-predicted increase in cancer risk. Based on all the above other consideration, any exposure to an impurity is later identified mutation is not necessarily associated with the increased cancer risk for patients already exposed to the impurity. The risk assessment would determine whether any future action will be taken. Then, where the proportional risk has been identified for impurity, an appropriate control strategy leveraging process understanding the analytical control should be developed to ensure that the mutagenic impurity is at below the acceptable cancer risk level. There may be cases where impurity is metabolic of the drug substance. In such cases, risk assessment to additional mutagenicity of metabolic can qualify the impurity. Consideration for marketed product. This guideline is not intended to apply retrospectively to products marketed prior to the adoption of this guideline. However, some types of post-approval changes warrant to be reassessment of safety related to mutagenic impurities. This section applies to those approval changes for products marketed under the after the adoption of this guideline. The life cycle one contains the additional recommendation for products marketed for adoption of this guideline. Post-approval changes of the drug substance chemistry manufacturing controls. Post-approval submissions involving the drug substance chemistry manufacturing controls should be include the evaluation of potential impact associated with mutagenic impurities from the changes to the route of synthesis reagents, solvents, then process condition after the starting material and specifically changes should be certain criteria for existing mutagenic impurities. Re-evaluation of impurities not be impacted by changes is not recommended. For example, when only a portion of Manufacturing process change. Assessment of risk from the mutagenic impurities should be limited to whether any new mutagenic impurities are found change, whether any mutagenic impurities found during the affected step are increased, whether any non-mutagenic impurities from upstream steps are increased. Regulatory submission associated with such changes should describe the assessment as outlined in section 9.2. Changing the site of manufacturer of drug substance, intermediate, starting material, and changing new raw material supplier will not require a reassessment of mutagenic impurity risk. When a new drug substance supplier is proposed, evidence that the drug substance produced by this supplier using the same route of synthesis as an existing drug product marketed in the associated region is considered sufficient evidence to acceptable risk benefit regarding the mutagenic impurities as an assessment to this guidance is not required. If this is not in case, then assessment of the guideline is expected. Then post-approval change to the drug product chemistry, manufacturing controls. Post-approval submission in a drug product, change in the composition, manufacturing process, dosage form should include an evaluation of potential associated with any new mutagenic degradation products or higher acceptance criteria for existing mutagenic degradation products. If appropriate, regulatory submission would be include the updated control strategy, re-evaluation of the drug substance associated with the drug products which not are recommended expected, provided there are no changes in the drug substance. Changing the site of manufacturing of the drug product will not require assessment of mutagenic impurity risk. Changes to the chemical use and marketed products. Changes to the clinical use of marketed products that can warrant re-evaluation of the mutagenic impurity limits include significant increase in the clinical use dose, increase in the duration of use, in particular when mutagen impurities control above the lifetime acceptable intake of previous indication that may not longer appropriate to longer treatment duration associated with the new indication. For change in the indication from the serious life-threatening condition where higher acceptable intake just referred to an indication for less serious condition where existing impurity acceptable intake may not longer be appropriate. Changes to the clinical use of marketed product associated with a new route of administration and expansion to into patient population that include the pregnant woman and pediatrics will not warrant re-evaluation, assuming increasing the daily dose of duration of treatment. Then other consideration for marketed product application of this guideline may not be warranted to marketed products if there is a specific cause for concern. The existence of impurity structural parts others alone is considered insufficient to trigger follow-up measures, and that is a structuring short of concern. However, a specific cause for the specific concern should be relevant impurity hazard data. This class 1 class is generated after the overall control strategy specification for the method authorization or established. This new relevant impurity hazard data should be derived from high quality scientific studies consistent with the relevant regulatory testing guidelines with data reports readily available. Similarly, newly discovered impurity is known as a class 1 class 2 mutation that is present in market product should be cause of concern. In both cases, when applicant becomes aware of new information, the evaluation of this guideline should be conducted. Drug substance and drug product impurity assessment. Actual potential impurities that are likely to arise during the synthesis storage of new drug substance drug product storage of should be assessed. The impurity assessment in two-stage process. Actual impurities that have been identified should be considered as mutagenic potential. An assessment of potential impurity likely to present in the final drug substance is carried out, determining if further evaluation of mutagenic potential is required. Then synthetic impurities, actual impurity that includes observed in drug substance above the ICSQ3 reporting threshold. Identification of actual impurities is expected when the levels are identification outlined by the ICSQ3. It is acknowledged that some impurities below the identification threshold may also be identified. Potential impurities in a drug substance can include storage starting materials and then reagents, intermediates in the route of synthesis from the starting material to the drug substance. Then risk of carryover of drug substance should be assessed to identify impurities that are present in starting material and intermediate. Those impurities that are reasonably expected by products. The route of synthesis from the starting material to the drug substance has a risk of carryover may be negligible in some impurities. A risk-based justification should be provided for the time to synthesis, after which these types of impurities should be evaluated for mutagenic potential. For starting material that are introduced and late in synthesis, their drug substance potential regression products in the drug substance and drug product. But it to be confirmed that the drug substance drug product under long-term conditions that are primary packaging. Knowledge of relevant degradation pathways can be used to help guide discussion decision on the selection of potential degradation products to be evaluated for mutagenicity. That is formal degradation chemistry principle, relevant trace testing studies, then development stability studies. Actual and potential degradation products likely to be present in the final drug substance. Drug products should be where structure is known should be evaluated for mutagenic potential described in section 6. Consideration of clinical development given in this chapter. Then hazard assessment elements given. Hazard assessment elements should be initiated and analysis actual potential impurities by the conducting database literature such as for carcinogenicity, bacterial mutagenicity data in order to classify them in a class 1 to 3 according to table 1. If data such classification is not available, the assessment structure activities are that is focuses on the bacterial mutagenicity prediction should be determined predicted that should be classified into class 3, 4, 5. Then impurities classification with respect to mutagenic and carcinogenic potential and resulting control action. Non-mutagenic carcinogens control are below the compound physical acceptable limits. Non-mutagenous what is the unknown carcinogenic potential. Then control of below the acceptable limits. Then altering structure and unrelated to the structure to the drug substance. No mutagenicity data. Control of below the acceptable TTC has control of bacteria mutagenicity, that is if normal mutagen class 5 is mutagen class 2. Altering structure is some alert in the drug substance and compound related to drug substance, that is treat on a non-mutagenic impurity. No structural alerts and electric structure with sufficient data demonstrate lack of mutagenicity cause and treat as a not mutagenic impurity. Then the substance structural errors from the two complementary QSR Saturn should be based on is sufficient to conclude the main impurity is not mutagenic concern. No further testing is recommended. As a class by if warranted, the outcome of any computer system based analysis should be reviewed by the use of expert knowledge of the not the additional supportive evidence or relevance of the positive negative complicate and the inclusive prediction provide the rationale for support the final conclusion. Then follow-up of the relevant structure or the declaration either adequate control measures should be applied or bacterial mutagen strategy should be conducted. Then risk categorization. As a result of hazard assessment described in a section 6, each impurity should be assigned to the one five class, that is the five impurities belonging to class 1 to 3. The principal risk categorization do derived acceptable intakes described in the section. TTC based acceptance intake. TTC-based acceptable intake mutagenicity impurity of 1.5 gram per person per day is considered associated with the negligible risk and can be generally used to the pharmaceutical as a default derived in acceptable limit for control. This approach should be usually used for